So, good morning everyone. Am I audible? Visibility is there, sound is coming. Audio, video, all okay? Right. So, probably today we have to start with the structure of cilia and flagella. Is it right? We have to start with what? Structure of cilia and flagella. Is it all right now? Basic idea I have given na in cilia and flagella. What are the difference between cilia and flagella? Right. So we have to talk about the structure, ultrastructure of cilia and flagella. And as I told you, both have the same ultrastructure. Both cilia and flagella have same ultrastructure. So let me explain how it look. So I'm just drawing here first the axoneme. So in axoneme, what is I'm making first? First I'm making central microtubule. So it is C1, this is C2. And this is which one? Cross bridge. This is which one? Cross bridge. You can say about it is C1, C2 bridge. C1, C2 bridge. Getting the things or not? Now what happened? These C1 and C2 is surrounded by, it is surrounded by which structure? Central sheath. Make it smaller somehow. It is surrounded by which one? Central sheath. So these structure is C1, C2, C1, C2 cross bridge, and this is what basically central sheath. This is central sheath. Got the idea? This is central sheath. Central sheath. Clear? Now what happened? 9 peripheral doublet is there. If you want to match your diagram like NCERT, you can do like this. Otherwise, it is circular, no all broken. This is central sheath. Now I am just making how many peripheral doublet? 9 peripheral doublet. 9 peripheral doublet. So just I have drawn a outline. Look here, I am just making peripheral doublet. So suppose that it is one doublet, it is second doublet, it is third doublet, right? Here it is fourth doublet, then it is fifth doublet, right? Then I am drawing here, assume sixth doublet. Here assume seventh doublet. This is assume here which doublet? Eighth doublet. So 1, 2, 3, 4, 5, 6, 7, 8. One doublet we have to make more. You can do like this. It is suppose that ninth doublet. So how many doublet are there? Ninth doublet is there. Starting from here 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 peripheral doublet are there. And each doublet you better know made up of which one? A, B. AB, microtubule. Got the idea? A and B is there. So what you will find, each A of a doublet joined with B of the another doublet with a linker called AB linker. So this is a protein whose name is AB linker protein. This AB linker protein is joining which one? A with B. A with B. Able to understand or not? So this is the AB linker. This is AB linker protein. It is AB linker protein. Got the idea? But with every A, there is arms is arising. With every A, arms is arising. It is inner arm, it is outer arm. Inner arm, outer arm. Inner arm, outer arm. Inner arm, outer arm. Inner, outer. Getting the things or not? Here, inner arm, outer arm. Inner, outer. Inner, outer. Inner, outer. So, inner arm is inside of the AB linker, outer arm is outside of the AB linker. Getting the idea or not? So, two type of arm is there, inner arm and outer arm. Getting the idea or not? So, here it is arms. Arms is there. Now with every A, radial spokes arising and move toward the central sheath and join with the central sheath. How many radial spokes? With every A, so how many A is there? 9 A is there, so 9 radial spokes are there. So this is what basically radial spoke. This is what basically radial spoke. So total 9 radial spokes are there. So I am just writing it is central singlet. It is what basically cross bridge. Got the idea? It is peripheral doublet. Peripheral doublet. So how many peripheral doublet is there? 9 peripheral doublet is there. How many central singlet is there? 2 central singlet is there, and that is the reason we are telling which arrangement? 9 plus 2 arrangement. Which arrangement? 9 plus 2 arrangement. So this is the structure of a exoneum. This is the structure of a exoneum. So label it also, it is central sheath. Central sheath. Now who is there? Plasma membrane is there. This central sheath is surrounded by periplasmic membrane. This is an exoneum structure. And when these exoneums surrounded by which one? Plasma membrane, it is called cilia or flagella. Getting the things? So it is which one? Periplasmic membrane. Periplasmic membrane, or you can also say it as what? Plasma membrane. Got the idea? Periplasmic membrane or plasma membrane. Is it clear in the mind? Is it clear in the mind? So this is the structure of what basically? Ultrastructure of cilia and flagella. Now I am telling you which protein is present where. It is important to know. So peripheral doublet is made up of tubulin protein. Tubulin protein is there in the peripheral doublet. If you talk about AB linker, which one? AB linker. Here it is made up of nexine protein. AB linker made up of nexine protein. Arms is made up of dynein protein. Dynein protein. And central singlet is also made up of dynein protein. So which protein is there? You got the idea. AB linker have nexine protein, right? Arms have dynein protein. Peripheral doublet have tubulin protein, and central singlet also have dynein protein. So at two places dynein protein is there. In entire doublet, who is there? In entire doublet, who is there? Tubulin protein is there. Now many times examiner will ask, and you will find rarely they ask the name of all protein. So usually what they ask? Which protein is present in cilia and flagella? And most of the time answer is what? Tubulin protein. You will find the exposure of such question where answer is what? Tubulin protein. So if tubulin, dynein, nexine, all of those is coming as an option, go with all of these. If many protein is there and tubulin is the option only, go for tubulin. Got the idea? How to handle the question with the name of protein. So which protein is present in cilia and flagella? So if nexine, dynein and tubulin not in option, suppose that flagellin is there, tubulin is there, pillin is there, all of these is there. So you will choose there which one? Tubulin protein. Got the idea? Picture came in the mind. So cilia and flagella is over. Getting the things or not? Now I am telling one more line which is important and question going to ask from here. Cilia and flagella arise from centriole-like structure called basal bodies. Cilia and flagella arise from which one? Centriole-like structure called basal body. Is it all right? Is it clear? Right. Now we are moving ahead. Clear? Done? Now we are moving ahead. Give your attention. Now we are going to deal about which one? Centrosome. So where centrosome is present? It is present in animal cell and few lower plant. Getting the things? Few lower plant. That is in almost all plant, centriole is absent. Centrosome or centriole is absent. Centrosome and centriole is absent. Got the idea? Almost all means not all. Almost all means someone are there who are present also. Almost all doesn't means all. Almost means usually, mostly. Now I am going to talk about the structure of the centrosome. So if you talk about a centrosome, in a centrosome, two centriole present perpendicular to each other and they are present near the nucleus. This centrosome in cell present where? Centrosome present near the nucleus. I am writing. Centrosome present near the nucleus. And if you talk about this centrosome, it have how many centriole? Two centriole perpendicularly placed to each other and they are surrounded by a amorphous pericentriolar material. So it is what basically? Amorphous pericentriolar material. amorphous pericentriolar material is there, and this is what basically 2 centriole. So it is a non-membranous structure. The amorphous pericentriolar material is not a what basically membrane. So this is a structure of centrosome. So a centrosome have what? 2 centriole which is surrounded by amorphous pericentriolar material. So this is the structure of centrosome. So you have seen the structure of a centrosome. Now if we go for the ultrastructure, which one? Ultrastructure of a centriole. If we go for a transverse section of a centriole, TS of a centriole, so we are getting which one? One centriole. I am taking a centriole and cutting the TS. So we are getting here 9 plus 0 arrangement. Which arrangement is there? 9 plus 0 arrangement is there. So 9 plus 0 arrangement is present in centriole. What the idea? And it have cartwheel like a structure. Who have a cartwheel like a structure? Centriole have a cartwheel like a structure. What the idea? So first you got the difference between centrosome or centriole not. Centrosome is a cell organelle, have how many centriole? 2 centriole. Getting the idea? It is a cell organelle, have how many centriole? 2 centriole, and both centriole are perpendicularly placed to each other, and they are surrounded by which one? Pericentriolar material, which is not a true membrane. So in centriole, who is there? It is membrane bounded organelle or no membrane bounded organelle? So cell membrane is absent, na? In centrosome and centriole, cell membrane is absent. Cell membrane absent. Are you getting the idea or not? Clear in the mind? So now we are going to talk about these 9 plus 0 arrangement of centriole, ultra structure under electron microscope, and it is a cartwheel like a structure of the centriole. Why we are telling it 9 peripheral, 9 plus 0 arrangement. So 9 who is there? Peripheral triplet is there. 9 peripheral triplet is there, and these are A tubule, B tubule, C tubule. So A, B and C, 3 tubules are forming a triplet, and such type of 9 peripheral triplet is present in the diagram of which one? Centriole. Now I am drawing the diagram of the centriole. Give your attention. So it is centriole. Or same diagram, who have? Basal body. So basal body and centriole have same ultrastructure. Basal body and centriole have same ultrastructure. I am just telling you, first you try to understand. This is a cell. Assume this is a cell. This is a cell. And from the cell, who is arising? Cilia and flagella is arising. Who is arising? Cilia and flagella is arising. Just try to understand. You will got a better picture. So it is like what? You will find like cilia and flagella when arising from a cell. Cilia and flagella when arising from a cell. So story is like this. You just try to understand. So suppose that it is cell membrane. So what is this? It is basal body. And this is what? Filament. And the part who is connecting the filament and which one? Basal body is what? Hook. Getting the thing, so now. So what you are finding, these basal body give rise to which one? Exposed part of cilia and flagella. Na? These basal body give the exposed part of the cilia and flagella. Na? Able to understand? Clear in the mind or not? These basal body give rise to the exposed part of the cilia and flagella. So that is the reason it is written like cilia and flagella arise from basal body. Actually, when you find basal body is a part of cilia and flagella, how many part is there? 3 part of cilia and flagella: basal body, hook and filament. So basal body is the part of cilia flagella, but when anybody writing cilia and flagella arise from basal body means they want to say what? Ki basal part of the cilia and flagella is expanding the which one? Extension. Like which one? Who is outside, visible part. It developed from which one? Basal body. Understanding the idea? So now I am going to tell you about the ultrastructure of which one? Centriole and basal body. So let me draw the diagram. Once again, we are going for a rough sketch diagram. So suppose that now I am going to make 9 peripheral triplet. So you think like it is one triplet. It is another 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 triplet. 1, 2, 3, 4, 5, 6. Suppose that it is another triplet. It is another triplet. Make it somehow far. It is another triplet. It is another triplet. It is one more triplet. So here I am making one more triplet. Wait a minute. One more triplet. So how many total triplet are there? Total 9 triplet are there. So in all 9 and in center, who is there in centriole structure? A hub is there. It is called central hub. It's somehow smaller. It is central hub. It is called central hub. And this is 9 peripheral triplet. 1, 2, 3, 4, 5, 6, 7, 8, 9. Now every triplet have 3 microtubule. And these 3 microtubule is what? A and B and C. So every A connected with the C. Every A connect with the other C. One and another triplet are connecting together, but how? A connect with the C. Who connect with whom? A connect with the C. And when A connect with the C, it is forming a such kind of a structure. Understanding the idea? So it is called which linker? AC linker. It is called AC linker. And this in center, it is no any microtubule. This structure is called what? Central hub. It is called central hub. Hub, H-U-B. Central hub. Got the idea? Now what happened? From every, below the every triplet, there is a dense material is there. And that material is called X bodies. So I am drawing here the diagram of X body. X body. And these X body, each X body attached with which one? A microtubule. Here A, B, C na. So each X body joined with which one? A microtubule of the triplet. X body is joined with A microtubule of the triplet. Later on what happened? Between 2X body, Y body is there. Between 2X body, Y body is there. So it is Y body. I am just giving the labeling. It is which body? Y body. And who is also interconnected? X and Y body are interconnected. Who are interconnected? X and Y body are interconnected. So I am just connecting which one? X and Y body. Give your attention. So here X body and Y body are interconnected like this. Getting the things or not? X body and Y body are interconnected like this. Now with every X body who is arising, radial spokes are arising. And these radial spokes are attached with which one? Hub. These radial spokes attached with which one? Hub. So here these are the radial spokes. From nine radial spokes will arise and they will join with the hub. So I am giving the labeling. It is which one? Radial spoke. Radial spoke. Got the idea? And this is which one? Peripheral triplet. Peripheral triplet. So when you look this structure, how does the structure look in appearance? It look like na cartwheel like a structure. Cartwheel like a structure. And this cartwheel structure is what basically? Centriole. So both basal body and centriole have same kind of arrangement. Which arrangement? 9 plus 0 arrangement. 9 plus 0 arrangement. Are you getting the idea or not? So this is the ultra structure of a centriole. I hope you understand it. This is the ultra structure of centriole. You got the idea? Picture came in mind? Is it done? Understood? So this is the ultra structure of centriole. 9 peripheral triplet connected by AC linker, central hub. From every A, X body connected, and through that radial hub is connected, radial spokes is connected with which one? Hub. And it is giving a which one? Cartwheel like a structure, and this structure is called centriole. Understanding the idea? So now I am writing the function of centriole. I am writing the function of centriole. So first function is what? It forms the basal body of cilia and flagella. Second function is spindle formation. Spindle formation. And this is a structure of a which one? Centriole. It is a structure of transverse sectional of which one? Centriole. Under an electron microscope. It is looking like this. So who is over? Centrosome and centriole is over. Centrosome and centriole is over. It is done. Now we are starting with which one? Nucleus. Can we proceed? It is done. Cilia, flagella, centriole, everything came in your mind. Understanding came? Or any problem, any glitch is there, any places? Understanding came or still any problem is there? Done? Clear? Yes. Now we are moving ahead. Now I'm starting with nucleus. So if you talk about the nucleus present in most eukaryotic cell. Most eukaryotic cell. It is first observed by Robert Brown. First observed by Robert Brown, and material inside of nucleus is stained, means colored by basic dye and who did it? By Fleming. And he called it chromatin. He called it chromatin. Got the idea? So now we are going to talk about number of nucleus in a cell. So number of nucleus we are going to discuss. Usually each cell have one nucleus. Usually each cell have how many nucleus? One nucleus. In some cells, more than one nucleus may present. One nucleus are also present. Example I can write paramecium. Paramecium have how many nucleus? Two nucleus. Got the idea? Another example you can say tapetum, that also have more than one nucleus. Another condition is what? In some cell, nucleus is absent. Is absent. An example I am writing mature mammalian RBC. Mature mammalian RBC. In mature mammalian RBC, who is absent? Nucleus is absent. Another example I am writing, sieve tube. Here also, nucleus is absent. Got the idea? So cell who perform more metabolic activity have large size of nucleus. So size of the nucleus is also depend upon metabolic function of the cell. So cell who are very active have bigger size nucleus. Understanding the idea? Now NCERT ask a question here. The cell where nucleus is absent, will you consider it living or dead? Pin your answer. Cell where nucleus is absent. An example, mammalian RBC, mature mammalian RBC, sieve tube. Will you say them living or dead? Sieve tube is dead. Mammalian RBC, your RBC is dead. Not, na? It is living. Who control the sieve tube? Nucleus of which one? Companion cell. You forget the anatomy? There I told or not told? Is it clear in the mind? Is it all right? Done. So now we are going to talk about the structure of nucleus. So number over, right? Now we are coming on structure of nucleus. A structure of nucleus. If we talk about structure of nucleus, It is a double membrane bounded cell organelle. Double membrane bounded cell organelle. So I am drawing the structure. Have a look here. Suppose that this is a cell. Okay. Now I am drawing the nucleus. Suppose that here it is one membrane of nucleus. Here it is another membrane of nucleus. So both membrane are parallel or not? Tell me first. Both membrane are parallel or not? Yes or no? Pin answer. Pin your answer. It is parallel or not parallel? It is parallel. Got the idea? Now what NCERT wrote? But problem is what? These membrane is not continuous. They are not continuous. Not continuous means what? They fuse with each other at certain locations. So both the membrane, this parallel membrane is not continuously present. It is interrupted and fused with each other. Are you getting the idea? So this nuclear membrane is a continuous structure or discontinuous structure? It is parallel, it is clear. It is continuous or discontinuous? Right? C or D? Continuous or discontinuous? C for continuous, D for discontinuous. So it is discontinuous. Very good. It is discontinuous. Well done. Now, why it is discontinuous? So where discontinuity is there, who is present? Nuclear pore is present. So this is nuclear pore. This is outer membrane of nucleus. Outer membrane of nucleus. This is inner membrane of nucleus. Inner membrane of nucleus. And these are in between outer and inner membrane. Who is there? Perinuclear space. So these perinuclear space, you can say, is a barrier between inside and outside, na? Any membrane or perinuclear space, all are the barrier between which one? Inside of the nucleus and outside of the nucleus. It is acting as a barrier or not? So if you talk about perinuclear space, so it is how much in diameter? 10 to 50 nanometer. Perinuclear membrane is how much? 10 to 50 nanometer in the diameter. Now, what NCERT wrote, when you read NCERT wrote like, nuclear membrane, outer membrane of the nucleus is continuous with endoplasmic reticulum. Outer membrane of the nucleus is continuous with endoplasmic reticulum. It is ER, it is plasma membrane, it is outer membrane. It is continuous with which one? Endoplasmic reticulum. So can I write here outer membrane continuous with ER, continuous with which one? Endoplasmic reticulum and contain ribosome. Contain ribosome. Understanding the idea or not? Now this is the nuclear pore. What nuclear pore is doing? These nuclear pore exchange RNA and protein in both direction. Both direction means what? Both direction means from inside of the nucleus to outside and from outside of the nucleus to inside. So material exchange, especially which one? RNA and protein exchange occur between cytoplasm and nucleus with the help of these nuclear pore. And these nuclear pore interrupt the plasma membrane, which one? Nuclear membrane. These nuclear pore interrupt the nuclear membrane. Understanding the idea or not? Now, if you look inside of the nucleus, you will find fluid-filled matrix present in the nucleus, and this fluid-filled matrix is called nucleoplasm. Nucleoplasm. Inside of the nucleus, nucleoplasm, two things is there. One here I am writing nucleolus, and another things I am telling chromatin network. Two things is present inside the nucleoplasm, fluid-filled matrix of the nucleus. So one I am drawing, it is nucleolus. So it is centrally located, made up of dense material. This structure is called nucleolus. This is called nucleolus. Getting the things or not? So another structure is present, which one? Chromatin network. So I will tell first of all about nucleolus, then I will even draw which one? Chromatin network. So nucleolus is what? Dense material inside of nucleus. You keep in your mind. Second important things you know about it is non-membranous. Who is non-membranous? Nucleolus is non-membranous. And that is the reason NCERT wrote it is connected with which one? Nucleoplasm. It is connected with rest of nucleoplasm. Because who is not there? Boundary is not there. Rest of nucleoplasm. If any boundary is not there, so it is continuous, na? So it is continuous with, I am writing the NCERT language. It is continuous with. You can consider continuous with as connected. Means nucleoplasm is touching with nucleolus. Able to understand or not? With the rest of the nucleoplasm. Getting the things or not? Clarity came in the mind. So it is made up of, I am telling about nucleolus, made up of ribosomal RNA and protein. Who is made up of ribosomal RNA and protein? Nucleolus. If you talk about nucleolus, its number is either 1 or more than 1. More than 1. It is not necessary every cell have only one nucleolus. When one and more than one, it depend on physiological state or metabolic activity of cell. Metabolic activity of the cell. So about nucleolus, what I told here, look here, I told it is a dense material inside the nucleus. It is non-membranous structure. It is continuous with the nucleoplasm. no boundary is there. It is made up of rRNA and protein. Its number is either one or more than one, depend upon the metabolic activity of the cell. Got the idea? This is about the nucleolus. Now I am coming to deal the function of nucleolus. Function of nucleolus. So what NCERT wrote about function? They wrote it is the site for synthesis of, it is the active site for, it is the active site for synthesis of ribosomal RNA. This is the line about nucleolus written in NCERT. It is the active site for synthesis of ribosomal RNA. Another function I am telling you, it is also called ribosome factory. Ribosome factory. Assembly of ribosome occur in which one? Occur in which one? Which one? Nucleolus. So you got the function of nucleolus. NCERT not wrote about this second function. NCERT only wrote about the first function. And what function they wrote? It is the active site for synthesis of ribosomal RNA. But I added a line. It is also called which factory? Ribosome factory, because ribosome get assembled at this place. Who get assembled at this place? Ribosome get assembled at this place. Understanding came in the mind or not? So now we are coming on chromatin. What we are going to discuss? Chromatin. Can we talk? Have a look. So I told na, nucleoplasm have two components. One component is nucleolus, we already discussed. Another component is chromatin network. And don't forget, these all structure we are talking about which nucleus? Interphasic nucleus. Interphasic nucleus means nucleus in interphase stage of the cell cycle. Because when cell undergoes in karyokinesis, after prophase 1, who disappear? Nucleus degenerate, disintegrate. Able to understand? Nucleus is not there. You studied na, nuclear membrane, nucleolus disappear in late prophase. So these are about which nucleus we are going to deal? Interphasic nucleus. What are the second component present in the nucleoplasm? Chromatin network. How it is present? First try to understand with the help of a diagram. First try to understand with the help of a diagram. Give your attention. So it is present like this. Got the idea? So this structure is what basically? Chromatin network. Got the idea? Clear in the mind? So what is this chromatin? I am just telling you about chromatin. It is extended, elaborate network of nucleoprotein present in interphase stage inside of nucleus. You got the idea? So it is an extended, elaborate network of nucleoprotein. And but these chromatin we find in which nucleus? Interphasic nucleus. Now I am asking a common sense based question. If I remove your body single cell, try to understand. If I remove a single cell from your body, and then I remove the nucleus, I will find who is there in the nucleus. Chromatin network is there, na? So in that chromatin network, how many chromatin is there? Pin your answer. How many chromatin is there? I am waiting for your answer. How many chromatin is there in the chromatin network? One answer came infinite. One answer came 1. Now, before answering, I am asking another question. Then I will give the answer, don't worry. How many chromosome is present in your somatic cell? Answer it. If I remove a cell from your body and I want to count the number of chromosomes, how many chromosomes is there? So now answer start coming correct. Sir, 46, 23 pair. Okay, very good, well done. So 46 chromosome is there. Getting the things. Do you know what is the difference between chromosome and chromatin? I am telling you. What is this? Chromatin. What is this? Chromatin. Understanding the idea? These chromatin is made up of nucleoprotein. So what is present in the chromatin? Nucleic acid and protein. Which nucleic acid? DNA, RNA. Which protein? Histone protein. Non-histone protein. So if someone asks you the composition of chromatin, what you will say? What is present in chromatin? DNA, RNA, histone protein and non-histone protein. These all together join and form chromatin. How it form, I will tell you in the molecular basis in second year. But you got the idea? Chromatin is made up of how many chemical compositions? DNA is there in chromatin, RNA is there, histone is there, non-histone protein is there. And how it is looking? It is an extended, elaborative network of nucleoprotein. Can I say it is a chromatin? Picture came or not? Composition of chromatin came. Composition of chromatin is what basically? DNA, RNA, histone protein, non-histone protein. Now I am asking a question. How many DNA present in one chromatin? It is a one chromatin. How many chromatin is this? One chromatin. How many DNA present in one chromatin? Chromatin. Answer of this question is 1. You will understand it much better when I will teach you nucleosome model. So you can make a formula that is what? 1 DNA at the rate of each chromatin. Understanding the things? So what is this? It is a chromatin. It is made up of what? DNA, RNA, histone protein, non-histone protein. How many DNA? 1 DNA. How many DNA? 1 DNA. How it look? It is an elaborate extended network of which chemical? Nucleoprotein. Picture came? Chromatin, understand? Now, what is the difference between chromatin and chromosome? These chromatin condense. These chromatin condense during prophase and metaphase, and maximum condense in which stage? Metaphase. So who condense? Chromatin condense. And after condensing, how it look, I am drawing the diagram. So I am going for condensation. Look now, this structure going for which one? Condensation. How it is looking, I am telling you. So it become like this. After condensation, it become like this. This is become like this after condensation. And this structure is called what? Chromosome. Or it is also called chromatid. Or better to write chromosome with one chromatid. So if I say how many chromosome is this, your answer must be what? 1. If I ask how many chromatid is this, your answer must be what? 1. Got the idea? Clear in the mind? Right? So in this chromatid, how many DNA is there? One DNA. In this chromosome, how many DNA is there? One DNA. Only differences is what in chromosome and chromatid. It is uncondensed, it is condensed. When it will uncondense, it become like this. When it become condensed, it become like this. So now I am asking a question. You told na, sir, in my somatic cell, 46 chromosome is there. Now I am asking then how many chromatid is there? Now answer the same question. How many chromatid is present in your somatic cell in interphase stage? How many chromatid? Answer it. 46 chromatid, na? 46 chromatid converted into 46 chromosome. Each chromatid converted into one chromosome. Understanding the idea? So if 46 chromosome is there, means how many chromatid is there? 46 chromatid is there. So just try to understand. Here it is a nucleus. And here, look, one chromatid, another chromatid, another chromatid, another chromatid, another chromatid, another chromatid, another. Many are present like this, and it is going to make a network called chromatid network. So in this network, how many threads are there? 46 threads are there. And when these 46, when you remove from this network, how many threads will come in your hand? 46 thread will come in your hand. And this each thread is called what? Chromatin. Chromatin. Understanding the idea? Each thread is called chromatin. Picture came. So in your somatic cell, at interphase stage, how many such thread is there? 46 such thread are there. Understanding the idea? How much such thread are there? 46 such thread are there. So how many DNA is there? If it is 46 thread, how many DNA is there? Tell me number of DNA. Tell me number of DNA. 46. Because we already told every chromatid have one. Every chromatin have one DNA. Every chromatin have one DNA. So 46 chromatin have how much? 46 DNA. Now I am telling length of human DNA. How much is it? Write your answer. In this chapter, NCERT wrote approximately 2 meter. It is written or not? Written, na? And in molecular basis of inheritance, it is written 2.2 meter. So 2.2 meter is exact. And 2 meter is approx. In cell structure, they wrote approximately 2 meter. In molecular basis of inheritance, they wrote the exact size, that is 2.2 meter. Understanding the idea? Now I am asking a question. It is the length of how many DNA? It is the length of how many DNA? 2 meter is the length of how many DNA? Answer it. 2 meter is the length of one student asking, sir, 1 DNA. Another student is saying, sir, 46 DNA. What are the contribution of another people? Pin your answer. 1 DNA, 2.2 meter, or 46 DNA, 2.2 meter. So the correct answer is 46 DNA. In every chromatin there is a DNA. In your interphysics somatic cell, 46 chromatin is there. They have 46 DNA. When I remove every DNA and place end to end, one DNA, then another DNA, then another DNA, then another DNA, then another DNA, all 46 when we plus like this, then which will come? 2.2 meter will come. Picture came or not? Understanding came or not? Is it clear in the mind or not? Tell me yes or no. Came. So 46 DNA have how much length? 2.2 meter. Now I am moving ahead. Give your attention. Have a look here. So many times you will say, sir, you are saying chromosome is this. But what I have studied, this is the chromatin, chromatid. It is chromatin and it is chromatid. What I study about chromosome is different from you. So you probably will say, I am just drawing your imagination of the chromosome. It will pakka match with your 100% match with your imagination. I am drawing your imagination of chromosome. Wait a minute. It is your imagination of chromosome, na? Uncondense, condense. It is your imagination of chromosome. If you are finding it is also not matching, so I am telling here, probably this must be your imagination. In your mind, chromosome, a structure is like this. Is it all right? Is it all right? Agree, understanding the idea? Now you can say, sir, I know this chromosome. You are telling this chromosome. What is correct? Both of us are correct. This is also a chromosome. This is also a chromosome. So here chromosome with how many chromatid is this? One chromatid, and this structure is what basically? Chromosome with two chromatid. So chromosome with one chromatid is called monad, and chromosome with two chromatid is called dyad. Chromosome with one chromatid is called monad. So it is chromosome also, chromatid also. How many chromosome? One chromosome. How many chromatid? One chromatid. How many DNA? One DNA. Which condition is this? Monad condition. When this will convert into this in S phase? In S phase what happened? Chromosome duplication occur. DNA replication occur. Formation of sister chromatid occur. Are you getting the idea now? So this one and this one are which one? Sister chromatid. So these sister chromatid formed during which one? S phase of the cell cycle. Duplication occur of the chromosome. Understanding the idea? So you got your dream chromosome. This is your dream chromosome now. Are you getting the idea? What is the how this is converting into this? One more copy is forming during which phase? S phase. And it is called which chromatid? Sister chromatid. Now you can say then why you are not telling here 2 chromosome? Here also how many chromosome is there? 1 chromosome. Here also how many chromosome is there? 1 chromosome. You can ask a question, na? Yeah, sir, you are not writing here 2 chromosome. Do you know why I am not writing 2 chromosome here? Do you know the reason why I am not writing? Because they are still connected. If they will separate, I will say 2. But they are still connected. Connected or not connected? Tell me. Tell me, connected or not connected? Connected, na? If I break this pen, right now how many pen is there? 1 pen. You will say, I am breaking the pen into 2 pieces. But 2 pieces not completely came. It attached at one point. And after that, 90% broken. And I am asking, Bhaiya, how many pen is there? What answer will you give? It is, you assume it is broken by 90%. And I am asking, how many pen is this? You will say 1 or 2. Until unless it will not separate connect completely. It is called what? 1. So here still it is connected. When it will separate and become 2 separate, we will say 2. But as long as it is connected, we will tell how many chromosome? 1 chromosome. So this condition of chromosome is called monad. This condition of chromosome is called dyad. In this condition of chromosome, before S phase, 1 DNA is there. After S phase, in 1 chromosome, how many DNA is there? 2 DNA is there. You got the idea? You understand the idea or not understand the idea? Tell me yes or no. Then I will move ahead. Then I will go for the labeling of the chromosome. Just have a look now. Now I am going for labeling. So I am erasing this diagram for labeling purpose. I want a space. Look here. So this is called primary constriction. Or it is called centromere. Centromere. Primary constriction or centromere. So 2 sister chromatid joined together at centromere. 2 sister chromatid joined together at which one? Centromere. Picture came or not came in mind? 2 sister chromatid joined together at centromere. Now here it is another constriction. It is called secondary constriction. Secondary constriction. Understanding the idea, it is called which constriction? Secondary constriction. Now try to understand. The bulge out region above the secondary constriction here is called satellite. Satellite. It is bulge out part after secondary constriction. After secondary constriction. And secondary constriction have fixed location.